So let us consider one important factor that plays a very important role in enzyme activity. It is the concentration of the substrate. So as you increase the concentration, notice that the x-axis is the concentration of the substrate and this curve represents the speed of the reaction or the velocity or how quickly the reaction is proceeding. You have kept the enzyme concentration constant over here. Okay, so say for example you have taken 100 enzymes with you and you are slowly increasing the substrate concentration along with the x-axis. As you move towards the right of the x-axis, the concentration of the substrate is increasing. So what are you modifying here? What are you changing here? You are changing the substrate concentration. Say for example you increase the concentration from 10 and then you make it 20 and then you make it 30 and then you make it 40 and then you make it 50. So as you are increasing the substrate concentration, the substrates are going and binding to the enzyme. The enzyme concentration is constant, and when the substrate goes and binds to the enzyme, the reaction is proceeding, the velocity of the reaction is advancing. So this curve gives you the progress of the velocity of the reaction. So like I told you, as you keep increasing the substrate concentration, the velocity increases. Now the question is, why does the velocity reach a constant value here? Why does it reach a maximal value here? Why we are not why the velocity is not progressing above this? Because all the enzymes have become used up. How many available enzymes are there in the solution in the reaction mixture? There are 100. So you cannot expect the reaction to proceed beyond 100 because there are 100 enzymes which are already available in the reaction mixture. If you add 200 substrates, all the 100 enzymes have already become occupied. Therefore, the maximal velocity of the reaction represents that point, that time point in the reaction when all the 100 enzymes that you have taken are saturated. When I say saturated, all of them have become bound by substrate. So saturated, or I will say occupied, their active sites are occupied. So because all the 100 enzymes that are available in the reaction mixture have become occupied, have become saturated, the velocity cannot go any higher. So Vmax represents that time point in the reaction when all the 100 enzymes that are available have become occupied or have become saturated. Then what do you mean by Vmax by 2? In Vmax by 2, half of the enzymes have become occupied. So say out of 100 enzymes, 50 enzymes have become occupied, 50 enzymes have become saturated. So when the velocity of the reaction reaches a point where half of the available enzymes have become saturated, that time point or that velocity is called half of the maximal velocity, or Vmax by 2. And you should find out what was the substrate concentration at that point of time. The substrate concentration which is necessary to ensure that half of the enzymes have become occupied by the substrate is referred to as the Km. So Km gives us the efficiency of the enzyme. Okay, it tells us how willing is an enzyme to go and bind to a substrate in order to make sure that half of the maximal velocity is attained. So please remember what exactly is Km. Km, see here they have extended the line from the curve to the x-axis. So Km actually talks about the substrate concentration, and here you can also extend a horizontal line towards the y-axis, which is nothing but the velocity of the reaction. So the substrate concentration required to attain half of the maximal velocity, that means out of 100 enzymes, 50 enzymes have to be occupied. That is referred to as the Km, and please note that Km is reflective of the efficiency of an enzyme. Lower the Km value, that is, if you get half of Vmax at a comparatively lesser concentration of substrate, that means the enzyme is more efficient. So think of it like this: you are keeping the substrate concentration, you are slowly increasing it, but you are keeping the enzyme concentration constant. Vmax represents that point in the reaction where all the enzymes are occupied. Maximal, you cannot have 101 enzymes being occupied because you only have 100 enzymes, and half of them, 50 enzymes, are occupied at Vmax by 2, and the concentration of substrate at that point of time is Km, or Km is actually also called the Michaelis-Menten constant, after those who worked out the dynamics involved in the effect of substrate concentration on enzyme activity.